Hello there, and welcome to the Dreamy Ocean Channel. As the day begins to gently dim around you and the world outside softens its insistent edges, perhaps you find yourself seeking a quiet harbor for your thoughts, a space where the mind can truly begin to unravel. Allow your body to unwind completely, to release any gentle burdens it may have carried throughout the hours. Let your shoulders drop your hands, rest peacefully, and feel the soft relaxation spread across your jaw and brow, softening the very lines of your face. With each slow, deliberate breath, feel a wave of tranquil calm ripple through you, inviting your mind to drift away from the day's concerns, allowing the distant noise to soften into a gentle, almost imperceptible hum. Tonight, we shall venture into the serene yet profoundly mysterious depths of our blue planet to contemplate a creature of surprising, almost ethereal beauty, and an even more astonishing secret, a being that embodies the ocean's profound paradox of life allure and lethal wonder. Let your mind float gently into these deep, dreamy waters, where our story will now softly unfold. The enigmatic guardians of the deep. Deep within the sun-dappled yet profoundly mysterious currents of our planet's vast oceans, there resides a creature of remarkable tranquility and astonishing silent power. Its form often plump and unassuming drifts with a slow, deliberate grace, a living testament to the gentle rhythm of the underwater world. This is the pufferfish, a member of the scientific family known as Tetraodontidae. A name that whispers of its unique dental structure, four fused teeth forming a formidable beak-like apparatus. Yet beyond this intriguing anatomical detail lies a deeper enigma, a secret held within its very being that has fascinated and at times tragically confounded humanity for centuries. Allow your mind to envision these subtle beings, perhaps a species like the tiger pufferfish. Fugu rubripes often found gliding amidst the temperate waters surrounding the islands of Japan. Their skin a canvas of soft browns and creams, patterned with delicate spots or subtle stripes, allows them to melt into the dappled light, filtering through the water's surface or to blend seamlessly with the sandy seafloor. Their movements are not those of a swift predator, but rather a series of gentle, almost hesitant undulations of their small, delicate fins. The dorsal and anal fins almost translucent flutter in a mesmerizing, synchronized motion, propelling the fish forward with a quiet dignity, as if conserving every droplet of energy, every fleeting moment of their existence. Their pectoral fins, positioned just behind their large, expressive eyes, allow for astonishing precision, enabling them to hover almost perfectly still, or to pivot with an effortless elegance that belies their somewhat bulbous form. Observe their eyes large and often independently moving like twin spheres of dark obsidian set into a soft, rounded face. They scan their surroundings with a thoughtful, almost contemplative gaze, absorbing the intricate details of their watery domain. The swaying fronds of seagrass, the textured surfaces of coral formations, the fleeting shadows of other aquatic life. There is a profound stillness in their observation, a patient watchfulness that speaks of an ancient wisdom, a deep connection to the ebb and flow of the ocean's lifeblood. They are not creatures of hurried impulse, but rather of deliberate, measured existence. Each small movement a considered response to the subtle cues of their environment. The most iconic and perhaps most captivating aspect of the pufferfish is its extraordinary ability to transform its physical self in an instant. When threatened, when a shadow looms too large or a predator approaches with too much intent, this seemingly docile swimmer undergoes a remarkable metamorphosis. It begins by rapidly ingesting water or even air if brought to the surface into a specialized, highly elastic stomach. Imagine this process unfolding in slow motion. The soft, pliable body, initially compact and streamlined, gradually begins to expand, swelling outwards from within. The scales, if present, or the small, often hidden spines embedded within its skin, become erect, transforming the smooth, unassuming surface into a formidable, prickly sphere. This inflation is a marvel of biological engineering, a testament to millions of years of evolutionary refinement. The pufferfish, by drawing in water, can increase its volume by several times, becoming a cumbersome, almost spherical object. This sudden increase in size serves two crucial purposes. Firstly, it makes the fish appear significantly larger and more intimidating to a potential predator, often deterring an attack simply by its sheer, unexpected bulk. Secondly, and perhaps more importantly, its inflated spherical shape makes it incredibly difficult for a predator to swallow. Imagine trying to consume a spiny, inflated ball. The task becomes nearly impossible, the risk of injury far outweighing any potential reward. The pufferfish, in this moment of vulnerability, becomes an impenetrable fortress, a living, breathing testament to the power of defense through transformation. As it floats fully inflated, its eyes, though still large, take on an almost comical yet intensely serious expression. The delicate fins which moments before propelled it through the water now seem almost comically small against its swollen body, fluttering occasionally as if to maintain balance in its new, ungainly form. It may drift helplessly with the currents, a living buoy, until the perceived threat has passed, and then with a slow, deliberate expulsion of the ingested water, it gradually deflates, returning to its original, more streamlined silhouette. This entire process, from tranquil swimmer to spiny sphere, and back again, is a silent ballet of survival, a testament to the ingenious strategies life employs to endure in a world of constant challenge. These creatures inhabit a diverse array of aquatic environments, their presence gracing the warm, vibrant coral reefs of tropical oceans, where they might gently graze on algae or pluck small invertebrates from crevices with their powerful beak-like jaws. They are also found drifting through the calmer, often murkier waters of estuaries, where fresh and salt water mingle, or even venturing into the expansive open ocean, though many prefer the protective embrace of coastal habitats. In these varied landscapes, they are often solitary figures moving with an unhurried dignity, seemingly content in their own silent company. Their diet primarily consists of various invertebrates, crustaceans, mollusks, and sometimes even smaller fish. Their unique, fused teeth are perfectly adapted for crushing the hard shells of crabs and clams, allowing them to access the tender flesh within a slow, methodical feast. Yet, it is not merely their remarkable ability to inflate, nor their patient demeanor that defines the pufferfish as an enigmatic guardian of the deep. There is another layer to their mystery, a profound and silent power woven into their very cells, a defense mechanism far more potent and insidious than any physical transformation. This hidden power resides within their tissues, a silent chemical secret that renders them not merely difficult to swallow, but profoundly lethally dangerous to consume. It is a secret that has been carefully guarded by nature's evolutionary hand, a chemical shield so potent that it places these seemingly gentle creatures among the most toxic vertebrates on Earth. This inherent toxicity is not a choice but an intrinsic part of their being, a legacy passed down through countless generations. It is a silent warning, a constant, unspoken declaration to potential predators. Approach with caution, For though I appear serene, I carry a hidden potency that can halt the very breath of life. The specific nature of this potency, its origins, and its profound effects remain a subject of deep scientific inquiry and a source of enduring fascination. It transforms the pufferfish from a mere inhabitant of the ocean into a living paradox, a creature of delicate beauty often observed with a sense of peaceful wonder, yet simultaneously a silent bearer of a lethal secret, an ultimate guardian of its own fragile existence protected by an invisible yet overwhelmingly powerful force. As the pufferfish drifts through its watery realm, perhaps nibbling on a piece of coral or gently sifting through sand for a hidden morsel, it carries this silent burden, this extraordinary gift of defense. It moves without fanfare, without aggression, a testament to the elegant simplicity of its survival strategy. Its existence is a quiet reminder that, in the depths of nature, even the most unassuming forms can harbor the most profound and astonishing capabilities. And so, these enigmatic guardians of the deep continue their slow, deliberate lives, their secrets held close, their presence a soft yet potent whisper in the vast, dreaming ocean. Tetrodotoxin, the silent architect of stillness, the silent, potent whisper carried by the pufferfish hinted at in the gentle currents of its deep-sea existence, is not a sound, but a substance, an intricate molecular architecture known as tetrodotoxin. This is not a venom injected with a fang or a barb, but rather a profound chemical secret held within the very tissues of the creature itself, a silent declaration of its formidable defense. Imagine a molecule so precisely crafted by the forces of evolution that a mere trace, a microgram quantity, almost imperceptible to the eye, possesses the power to quiet the most complex biological systems, bringing the vibrant symphony of life to a profound, irreversible stillness. Tetrodotoxin, often abbreviated as TTX, is a neurotoxin of extraordinary potency, a compound so exquisitely effective at its task that it stands among the most lethal non-protein natural toxins known to science. Its name itself derived from the family Tetraodontidae. The very pufferfish we contemplate links it inextricably to its most famous bearer. Yet this remarkable substance is not unique to these oceanic guardians. Its presence has been discovered in a surprising array of other organisms, including certain species of blue-ringed octopuses, newts of the genus Terecha, some vibrant marine snails, and even specific species of frogs, painting a picture of a potent chemical weapon, widely distributed across diverse evolutionary branches, each time serving as a silent, formidable deterrent. To truly grasp the astonishing power of tetrodotoxin, consider this: it is estimated to be approximately 1,200 times more potent than cyanide, a substance universally recognized for its swift and brutal efficacy. Imagine the difference between a gentle breeze and a hurricane. The destructive potential is on an entirely different scale. A single milligram of pure tetrodotoxin, a speck no larger than a grain of fine salt, holds enough power to potentially claim the lives of several adult humans. This potency is not a sudden, explosive force, but rather a quiet, insidious unraveling, a dismantling of the fundamental signals that orchestrate life itself. The story of tetrodotoxin, like many profound scientific discoveries, is interwoven with ancient observations and the slow accumulating wisdom of generations. For centuries, perhaps even millennia, coastal communities, particularly in East Asia, understood the dual nature of the pufferfish, a potential source of sustenance, yet simultaneously a harbinger of swift, silent death. Records from ancient Japan speak of the dangers of the delicate balance between culinary daring and fatal consequence. The precise chemical identity of the toxin, however, remained shrouded in mystery until the early 20th century. It was through meticulous scientific inquiry, a painstaking process of isolation and analysis, that researchers finally unveiled the molecular structure of this enigmatic compound, revealing its intricate arrangement of carbon, hydrogen, nitrogen, and oxygen atoms, a complex puzzle piece of nature's design. Within the pufferfish, tetrodotoxin is not uniformly distributed throughout the body. Its concentration varies significantly, not only between different species of pufferfish, but also within different organs of a single fish, and even seasonally. The highest concentrations are typically found in the liver, the ovaries, and the skin. The intestines, eyes, and sometimes even the muscle tissue can also contain varying, though generally lower amounts of the toxin. Picture this internal landscape, a living creature carrying pockets of potent poison, almost like specialized glands, meticulously sequestering this dangerous compound to ensure its own survival. This uneven distribution is a critical factor in the careful art of preparing pufferfish for consumption, an art form that demands an intimate understanding of these toxic geographies. A natural question arises from this fascinating arrangement. If the pufferfish harbors such a potent toxin within its own body, how does it avoid self-poisoning? The answer lies in a subtle yet profound evolutionary adaptation. The pufferfish, through countless generations of natural selection, has developed a remarkable resistance to tetrodotoxin. Scientists believe that this resistance stems from a slight yet crucial alteration in the molecular structure of their own sodium channels, the very targets upon which tetrodotoxin exerts its devastating effects in other organisms. Imagine a lock, and a key. Tetrodotoxin is the key perfectly shaped to fit and jam the sodium channel lock in most creatures. But in the pufferfish, the lock itself has been subtly reshaped, rendering the key ineffective, allowing the toxin to exist harmlessly within its system, a silent guardian that cannot turn against its host. This silent architect of stillness does not cause pain in the conventional sense. It does not burn or corrode. Instead, its action is far more insidious, far more fundamental. It operates at the very bedrock of nerve impulse transmission, targeting the delicate electrical signals that orchestrate every movement, every sensation, every breath. Its primary mechanism involves an astonishingly precise blockade of voltage-gated sodium channels in nerve cell membranes. These channels are like tiny molecular gates embedded within the outer surface of nerve cells, responsible for the rapid influx of sodium ions that generates an action potential, the electrical impulse that allows nerves to communicate. Imagine millions upon millions of these microscopic gates opening and closing in perfect synchronicity, allowing electrical messages to flow like gentle currents through the vast, intricate network of the nervous system. Tetrodotoxin, with its precise molecular structure, slips into the outer pore of these sodium channels, binding with an incredibly high affinity. It acts like a tiny, perfectly fitted plug, obstructing the channel and preventing the passage of sodium ions. When these gates are plugged, they cannot open and the electrical impulses cannot fire. The nerve signals cannot propagate. The consequences of this molecular blockade are profound and widespread. Without the ability to generate and transmit electrical signals, the nervous system begins to falter. Muscles which rely on nerve impulses to contract gradually lose their ability to respond. The diaphragm, the crucial muscle responsible for breathing, becomes paralyzed. The heart's electrical rhythm, though somewhat less affected initially, can also be impacted. The brain, though not directly targeted by TTX in a way that would alter consciousness, becomes isolated, unable to send its commands to the body, unable to receive sensory input from the world outside. It is a terrifying state of complete conscious paralysis, a stillness imposed from within, while the mind remains agonizingly aware. The origin of tetrodotoxin in the pufferfish is another layer of its profound mystery. For a long time, it was assumed that pufferfish synthesized the toxin themselves. However, scientific consensus has shifted. Current research strongly suggests that pufferfish do not produce tetrodotoxin intrinsically, but rather acquire it through their diet. They consume specific bacteria often found in the marine environment, or invertebrates that have accumulated these bacteria or other TTX-producing organisms. These bacteria, such as certain species of Vibrio or Alteromonas, are the true alchemists synthesizing the complex tetrodotoxin molecule. The pufferfish then absorbs and sequesters this toxin in its various organs, building up its internal chemical arsenal as it feeds. This dietary acquisition explains why pufferfish raised in controlled, toxin-free environments, fed on a diet devoid of TTX-producing bacteria, often remain non-toxic. It is a testament to the intricate food webs and microbial interactions that shape the chemical landscape of marine life. Thus, tetrodotoxin stands as a silent architect, a master of stillness. It is a molecule that whispers of the ocean's intricate chemistry, of evolutionary arms races, and of the profound vulnerability of life's most fundamental processes. It is a testament to nature's capacity for both exquisite beauty and astonishing, quiet lethality. A secret held within the gentle form of the pufferfish, waiting to be understood, to be respected, and to be contemplated in the quiet depths of our own awareness. Its story is one of molecular precision, of life's delicate electrical dance, and of the profound stillness that can descend when that dance is suddenly, irrevocably halted. Whispers in the nerves how tetrodotoxin silences life. Having gently unveiled the silent presence of tetrodotoxin, that intricate molecular architect of stillness, let us now drift deeper into the very mechanism of its power to understand precisely how it orchestrates that profound quietude within living systems. Imagine, if you will, the intricate shimmering pathways of the nervous system, a vast, silent network of communication that underpins every thought, every sensation, every deliberate and involuntary movement of a living being. Within this complex landscape, electrical signals known as action potentials are the fundamental whispers, the rapid pulses that carry information from one nerve cell to the next, from the brain to the muscles, from the sensory organs back to the processing centers. These action potentials are generated by the precise rhythmic opening and closing of tiny specialized proteins embedded within the outer membrane of every nerve cell, known as ion channels. Among these, the voltage-gated sodium channels are paramount for the initiation and propagation of these electrical signals. Picture them as exquisitely sensitive gates awaiting a specific electrical cue to momentarily swing open, allowing a rush of positively charged sodium ions from the outside of the cell to flood into its interior. This sudden influx of positive charge is the very spark, the initial surge that ignites the action potential, sending it propagating like a wave along the nerve fiber. Now, envision tetrodotoxin, that precisely structured molecule, as it enters the bloodstream after ingestion. It is absorbed with a quiet efficiency, making its way through the circulatory system, carried by the gentle current of the blood, until it reaches its ultimate destinations, the nerve cells and muscle cells throughout the body. Once it encounters a voltage-gated sodium channel, its true purpose begins to unfold. The tetrodotoxin molecule possesses a unique configuration that allows it to bind with an incredibly high affinity and specificity to a particular site within the outer pore of these sodium channels. Think of it as a perfectly crafted key slipping into a lock, but instead of opening it, this key jams the mechanism completely. As tetrodotoxin binds it, effectively plugs the channel, preventing it from opening. The molecular gate is frozen shut. No matter how strong the electrical stimulus, no matter how urgent the message attempting to pass through the sodium ions, cannot rush in. The critical influx of positive charge, the very spark of the action potential, is extinguished. The nerve cell, deprived of its ability to generate these electrical whispers, becomes silent. It can no longer communicate. This blockade is not a destructive force that tears apart the cell. Rather, it is a subtle yet profound interruption of function. The nerve cell itself remains structurally intact, its internal machinery still present, but its capacity to transmit signals, its very purpose, is utterly disabled. This is why the effects of tetrodotoxin are so insidious. The mind, the center of consciousness, may remain lucid and aware, trapped within a body that can no longer respond, a silent prisoner within its own paralyzed form. Consider the cascading effects of this neural silence. The first areas to be significantly impacted are often the peripheral nerves, those delicate fibers that extend from the brain and spinal cord to the extremities, carrying commands to muscles and relaying sensory information back. As the sodium channels in these nerves are progressively blocked, the ability to initiate voluntary muscle movement begins to fade. Imagine the subtle tremor in a fingertip, the slow weakening of a limb, the growing difficulty in lifting an arm or moving a foot. These are the early whispers of the toxin's presence, the initial signs of the body's communication system being gently yet firmly muted. Sensory nerves are also affected. The ability to perceive touch, temperature, and pain diminishes. The world outside begins to recede, not because the brain ceases to register it, but because the pathways for those sensations to reach the brain are slowly being closed off. A chilling numbness spreads, often starting around the mouth, and extremities are feeling akin to pins and needles, which then deepens into a profound, heavy insensitivity. This sensory paralysis adds another layer to the terrifying experience, severing the connection between the inner self and the outer world. The most critical and life-threatening consequence of tetrodotoxin's action is its effect on the muscles essential for respiration. Breathing is an involuntary act, yet it relies entirely on the rhythmic contraction and relaxation of the diaphragm and intercostal muscles, all orchestrated by precise nerve impulses originating in the brainstem. As tetrodotoxin silences the sodium channels in the motor nerves leading to these respiratory muscles, they lose their ability to contract. The diaphragm, the primary muscle of inhalation, becomes flaccid and unresponsive. The chest muscles, which assist in expanding and contracting the ribcage, also cease their vital work. Imagine the slow, agonizing realization as the breath becomes shallower, more labored, until it eventually ceases altogether. This respiratory arrest is the ultimate cause of death in tetrodotoxin poisoning. The heart, though it has its own intrinsic pacemaker cells that are somewhat less sensitive to TTX, can also be affected at higher doses or prolonged exposure, leading to arrhythmias or a complete cessation of its rhythmic beat. But often, it is the inability to breathe that precedes cardiac failure. A cruel irony where the heart continues to beat for a time, pumping blood through a body that is no longer able to draw the life-giving oxygen from the air. The brain itself, unlike the peripheral nerves, is protected to some extent by the blood-brain barrier. a specialized network of cells that carefully regulates which substances can pass from the bloodstream into the delicate neural tissue of the central nervous system. While some tetrodotoxin can eventually cross this barrier, its primary and most immediate devastating effects are on the peripheral nervous system. This means that for a significant period during the paralysis, the victim's consciousness often remains intact. They are fully aware of their surroundings, of their own body's horrifying descent into immobility, but utterly unable to move, to speak, to signal for help. This conscious paralysis, this terrifying lucidity within an unresponsive shell, is perhaps the most harrowing aspect of tetrodotoxin poisoning, turning a chemical event into a profound psychological ordeal. There is no known antidote to tetrodotoxin. Medical intervention focuses entirely on supportive care, a race against time to maintain vital functions until the body can slowly metabolize and excrete the toxin. This typically involves immediate gastric lavage to remove any unabsorbed toxin from the stomach, followed by aggressive respiratory support, often through mechanical ventilation. Imagine a machine gently, rhythmically breathing for a person, forcing air into and out of their lungs, keeping the essential exchange of oxygen and carbon dioxide flowing, while the body itself remains stubbornly still, unable to perform this most fundamental act of life. Intravenous fluids are administered to support circulation and aid in toxin excretion through the kidneys. The hope is that given enough time and meticulous support, the tetrodotoxin will eventually unbind from the sodium channels, be cleared from the system, and the channels will slowly regain their function, allowing the whispers in the nerves to gradually resume their vital communication. The recovery, if it occurs, is often a slow, gradual process, as the body painstakingly rebuilds its neural communication pathways. The first signs might be a subtle flicker of movement in a finger, a slight twitch of an eyelid, a whisper of a breath. Each small return of function is a profound victory, a testament to the resilience of life against the silent architect of stillness. This profound chemical mechanism operating at the subcellular level transforms the pufferfish from a mere creature of the ocean into a living embodiment of nature's potent chemistry. It is a testament to the astonishing precision with which evolution can craft a defense, a silent molecular shield that protects the pufferfish by silencing the very essence of life in those who would consume it. And so, the whispers in the nerves, the electrical dance of existence, are brought to a profound and terrifying halt by the subtle yet overwhelming presence of tetrodotoxin, a silent force operating at the very heart of cellular communication. The bacterial alchemists, tracing the toxin's genesis. Having explored the precise, devastating mechanism by which tetrodotoxin silences the intricate whispers of the nervous system, a deeper question now gently surfaces from the scientific depths. Where does this astonishing molecule truly originate? For many years, the prevailing belief was that the pufferfish, along with other toxic creatures, possessed the intrinsic biological machinery to synthesize tetrodotoxin within its own cells. It was a compelling idea suggesting an inherent, almost magical self-sufficiency in creating such a potent chemical defense. However, as the subtle currents of scientific inquiry continued to flow, meticulously gathering data and refining understanding, a more nuanced and profoundly interconnected story began to emerge, one that points not to the pufferfish itself, but to an even more ancient and ubiquitous form of life, the humble bacterium. Imagine a microscopic world teeming with countless forms of life, invisible to the unaided eye, yet profoundly influential in shaping the larger ecosystems around them. These are the bacteria, ancient alchemists of the earth, capable of performing an astonishing array of chemical transformations, synthesizing compounds that range from life-sustaining nutrients to potent toxins. It is within this unseen realm that scientists now largely believe the true genesis of Tetrodotoxin lies. The current scientific consensus, supported by a growing body of evidence, posits that pufferfish do not produce tetrodotoxin endogenously, but rather acquire it from external sources primarily through their diet. This hypothesis began to gain traction as researchers observed a curious phenomenon: pufferfish raised in laboratories, in environments carefully controlled to be free of tetrodotoxin-producing microorganisms, and fed a sterile diet, often remained non-toxic. They lacked the lethal potency of their wild counterparts, suggesting that something in their natural environment, something they consumed, was the source of the toxin. This observation was a pivotal whisper guiding scientists toward a new understanding. The trail led to the discovery of specific bacterial species, primarily marine bacteria, belonging to genera such as Vibrio, Pseudo-Alteromonas, Shewanella, and Alteromonas, among others. These microscopic organisms, thriving in the rich, complex ecosystems of marine sediments, algal mats, and even as symbionts within the guts of various invertebrates, possess the biochemical pathways necessary to synthesize the intricate tetrodotoxin molecule. Envision these tiny factories, each bacterial cell a miniature chemical plant, meticulously assembling the atoms of carbon, hydrogen, nitrogen, and oxygen into the precise configuration that forms TTX. The acquisition process for the pufferfish is believed to unfold through a complex food web. Pufferfish, being omnivorous or carnivorous, consume a variety of smaller organisms in their natural habitat. They might graze on algae or detritus, where these TTX-producing bacteria reside. More commonly, they feed on invertebrates such as crabs, starfish, marine worms, or snails. Many of these invertebrates in turn may have either consumed the bacteria directly or accumulated tetrodotoxin from their own diets, becoming vectors for the toxin. So a pufferfish might consume a small crab, and that crab might have previously consumed algae harboring toxin-producing bacteria, or even other small organisms that had accumulated TTX. It is a subtle chain of consumption, a silent transfer of chemical power, moving up through the trophic levels. Once ingested by the pufferfish, the tetrodotoxin is absorbed from the digestive tract and then selectively sequestered and concentrated in specific organs. This selective accumulation is another marvel of biological adaptation. The pufferfish's body is remarkably efficient at taking up the toxin and storing it, particularly in the liver, ovaries, and skin, without suffering harm itself. As we discussed, this resistance is attributed to unique modifications in their own sodium channels, rendering them impervious to the toxin's binding action. Imagine a highly specialized storage facility within the pufferfish, carefully collecting and holding this potent chemical, not for immediate use in an offensive strike, but as a constant internal shield, a deterrent that is always present. The concentration of tetrodotoxin within a pufferfish can vary dramatically based on several factors. The species of pufferfish plays a role, as some species are inherently more toxic than others. The geographical location is also significant, as different marine environments harbor different populations of TTX-producing bacteria and different food sources. Furthermore, the season can influence toxicity, with some species exhibiting higher toxin levels during spawning periods when the ovaries, a primary storage site, are enlarged and more active. Even the individual diet of a specific fish can dictate its toxicity. A pufferfish that has consistently consumed toxin-laden prey will naturally accumulate more TTX than one whose diet has been less exposed. This variability underscores the complexity and unpredictability inherent in managing the risk associated with these creatures. This understanding of the bacterial origin of Tetrodotoxin has profound implications extending far beyond the pufferfish itself. It suggests that the presence of TTX in other diverse organisms, from blue-ringed octopuses whose vibrant warning colors belie a swift, silent lethality to certain newts that secrete the toxin from their skin, and even some species of marine snails, likely follows a similar pattern of dietary acquisition from bacterial sources. This paints a picture of a shared chemical weapon independently acquired and adapted by various lineages across the tree of life, all thanks to the metabolic prowess of microscopic bacteria. The discovery of these bacterial alchemists also opens new avenues for research, not only into the biosynthesis pathways of tetrodotoxin itself, but also into the broader ecological role of these toxin-producing microorganisms. How do these bacteria benefit from producing such a potent compound? Is it a defense against predation by protozoa or other microbes? Is it a signaling molecule in their own complex microbial communities? These are questions that continue to gently ripple through the scientific community, inviting further exploration into the hidden lives of the smallest inhabitants of our planet. Moreover, the knowledge that pufferfish acquire their toxicity from their environment has practical applications, particularly in aquaculture. Scientists and aquaculturists have successfully raised pufferfish in controlled, toxin-free environments, providing them with diets free of TTX-producing organisms. These farm-raised pufferfish under strict conditions can be rendered non-toxic, offering a potentially safer alternative for culinary purposes. However, the rigorous controls required and the ever-present risk of accidental exposure to toxin sources mean that wild-caught pufferfish will always carry their inherent acquired potency, demanding the utmost respect and caution. So the next time you consider the pufferfish drifting silently in the ocean's embrace, remember the unseen forces at play. Remember the microscopic bacteria, those ancient alchemists quietly performing their chemical magic, synthesizing the potent tetrodotoxin. Remember the intricate food webs, the subtle transfers of energy and matter that carry this molecule from the smallest microbe to the larger marine creatures. And remember the pufferfish itself, not as the creator of its own deadly secret, but as a cunning collector and protector of a powerful chemical gift acquired from the vast, interconnected tapestry of marine life. It is a testament to the profound and often surprising ways in which life on Earth influences and shapes itself, a silent, ongoing chemical conversation beneath the waves. Fugu, a culinary dance with mortality. Having journeyed through the unseen world of bacterial alchemists and understood the silent, profound mechanism of tetrodotoxin, let us now turn our gaze towards a most extraordinary cultural phenomenon, one that dares to embrace this potent chemical secret as an integral part of a revered culinary tradition. This is the world of fugu, the Japanese term for pufferfish, and the exquisite, perilous dishes prepared from it. It is a world where gastronomic delight dances on the very precipice of danger, where meticulous skill and profound respect for life and death converge in a single delicate meal. Imagine a culinary landscape steeped in centuries of tradition, where the pursuit of flavor texture and aesthetic presentation reaches an almost spiritual level. Within this rich tapestry, fugu occupies a unique and revered position. It is not merely a dish, it is an experience, a statement, a testament to courage, precision, and an almost philosophical acceptance of risk. For those who seek it, the allure of fugu lies not just in its delicate, translucent flesh, but also in the subtle, almost imperceptible tingle it can impart upon the lips and tongue, a fleeting sensation that whispers of the potent toxin lurking just beneath the surface. A reminder of the chef's extraordinary skill and the diner's audacious trust. The preparation of fugu is not a mere act of cooking. It is a ritual, a precise surgical art form passed down through generations. The goal is to carefully remove all toxic parts, primarily the liver, ovaries, and intestines, while preserving the edible non-toxic muscle tissue and skin. Each cut of the knife is a deliberate act, a testament to years of training and an unwavering focus. The chef, armed with specialized tools and an intimate knowledge of pufferfish anatomy, must navigate the internal landscape of the fish with the precision of a surgeon, knowing that a single misstep, a tiny slip of the blade, could mean the difference between an exquisite meal and a fatal poisoning. The most prized part of the fugu is its delicate white flesh, often served as sashimi, known as fugu sashi or tessa. Imagine thinly sliced pieces, almost transparent, arranged with artistic grace on a large platter, sometimes in patterns resembling chrysanthemums, a symbol of longevity in Japan. The texture is firm yet subtly yielding, with a clean, understated flavor that is highly valued. It is often dipped in ponzu sauce, a citrus-based condiment, perhaps with a touch of momiji oroshi, grated daikon radish with chili pepper and finely chopped scallions. The experience is not about an overwhelming burst of flavor, but rather a subtle interplay of textures and delicate notes, a quiet appreciation of the pristine quality of the fish. Beyond the raw sashimi, fugu can be prepared in various other ways. Fugu chiri, or techiri, is a hot pot dish where the fish, along with vegetables and tofu, is gently cooked in a simmering broth. The delicate flavor of the fugu infuses the broth, creating a warming, comforting experience. Fried fugu, fugu karaage, offers a different texture, with the crisp exterior yielding to the tender, flaky flesh within. Even the skin, meticulously cleaned and prepared to remove all traces of toxin, can be served as yubiki blanched and thinly sliced, offering a chewy, gelatinous texture, often dressed with ponzu. There is also the peculiar tradition of hirazake, where a grilled fugu fin is steeped in hot sake. The smoky aroma and umami notes from the fin infuse the sake, creating a unique, warming beverage. This too is part of the fugu experience, a multi-sensory journey that extends beyond the flesh itself, embracing every carefully prepared part of the fish that can be rendered safe for consumption. The allure of fugu is multifaceted. For some, it is the sheer novelty, the thrill of engaging with a food that carries such a profound reputation for danger. For others, it is a deep appreciation for the chef's mastery, a respect for the extraordinary skill required to transform a potentially lethal creature into a work of culinary art. And for many connoisseurs, it is the unique flavor profile, the subtle texture, and the almost indescribable sensation that can accompany a perfectly prepared fugu meal. This sensation, often described as a gentle tingling or numbness on the lips and tongue, is not a sign of poisoning, but rather a carefully controlled, minute exposure to the toxin, meticulously balanced by the chef to enhance the experience without crossing the line into danger. It is this delicate balance, this art of controlled risk, that makes fugu dining so uniquely compelling. The history of fugu consumption in Japan stretches back millennia. Archaeological evidence, including fugu bones found in shell mounds dating back more than 2,000 years, suggests that people have been consuming pufferfish for a very long time. However, the dangers were always present. Historical records are punctuated by accounts of poisonings, even prohibitions. During the Edo period, for instance, the consumption of fugu was strictly forbidden, for samurai perhaps to prevent accidental deaths among the warrior class, whose lives were deemed too valuable to be risked on a meal. Yet despite these prohibitions and the known risks, the allure of fugu persisted deeply, embedding itself into Japanese culinary culture. The regulations surrounding fugu preparation and consumption in Japan are among the strictest in the world, a testament to the inherent danger of the fish. These regulations are not arbitrary. They are the result of centuries of experience and a profound understanding of the toxin's power. The system of chef licensing, which we will explore in greater detail, is designed to ensure that only individuals with the highest level of skill knowledge and ethical responsibility are permitted to prepare fugu for the public. This rigorous oversight is what allows fugu to be served safely in licensed establishments, transforming a potentially fatal meal into a carefully managed, revered culinary experience. So, as we contemplate fugu, we are not merely considering a dish, but a complex interplay of culture, science, risk, and extraordinary human skill. It is a culinary dance with mortality performed with grace and precision, a testament to humanity's enduring fascination with the boundaries of experience and a profound respect for the potent secrets held within the depths of the ocean. The delicate slices of tessa, arranged like petals on a plate, represent not just a meal, but a story, a tradition, and a quiet moment of profound trust between the diner and the master chef, a silent acknowledgement of the thin, shimmering line between life and stillness. The way of the fugu chef, precision in the blade's edge. From the refined allure of fugu as a culinary experience, our journey now gently shifts to the very heart of its preparation, to the hands that transform peril into pleasure. This brings us to the revered figure of the fugu chef, a master craftsman whose artistry is defined not just by flavor and presentation, but by an unwavering precision, a profound knowledge, and an almost sacred responsibility. The path to becoming a licensed fugu chef in Japan is not merely a course of study. It is an arduous apprenticeship, a rigorous trial by fire demanding years of dedication, and an absolute mastery over both the fish and oneself. Imagine the quiet intensity that pervades a licensed fugu kitchen. The air itself seems to hum with a focused energy, a silent testament to the gravity of the task at hand. The chef, often clad in pristine whites, approaches the fugu with a reverence born of deep understanding. Each fish is not merely an ingredient. It is a living entity, a vessel containing both sustenance and potential demise. The tools of the trade are carefully chosen, specialized knives, often with long, slender blades honed to an almost molecular sharpness, capable of making cuts of extraordinary delicacy and precision. These are not merely instruments. They are extensions of the chef's will, guided by years of muscle memory and an intimate anatomical knowledge. The first step in this meticulous process is the careful selection of the fish itself. Licensed establishments source their fugu from reputable suppliers, often from specific fishing grounds known for healthy, well-maintained fish. Upon arrival, each fugu is inspected, its species identified, as different species can have varying levels and distributions of Tetrodotoxin. This initial identification is critical, setting the stage for the specific preparation techniques that will follow. Once selected, the fugu is brought to the preparation area, often a dedicated space within the kitchen designed for sterility and absolute focus. The chef begins by making an incision behind the pectoral fins, a swift, practiced movement that severs the spinal cord, stunning the fish. Then, with a series of precise cuts, the fins are removed, followed by the careful removal of the skin. The skin itself, often toxic, must be peeled away with utmost care, ensuring no contact with the edible flesh. Imagine the steady hand, the unwavering gaze as the chef meticulously separates layers, each movement economical and deliberate. The true artistry and the greatest challenge lies in the evisceration. This is where the lethal organs reside: the liver, the ovaries in females, and the intestines. These are the parts that harbor the highest concentrations of tetrodotoxin, and their removal demands surgical precision. The chef must isolate each organ, carefully detaching it from the surrounding muscle tissue without piercing or contaminating the edible flesh with even a microscopic trace of the toxin. The liver, often a large, dark organ, is particularly dangerous as it can contain exceptionally high levels of TTX. The ovaries, especially during spawning season, are another critical point of focus, bulging with potential lethality. Each removed toxic organ is placed immediately into a dedicated locked container, often a metal bin clearly marked toxic or inedible. This container is then securely sealed and later disposed of according to strict regulations, often incinerated at a specialized facility, ensuring that the potent poison poses no further threat. This meticulous segregation is a testament to the profound respect for the toxin and the absolute commitment to public safety. After the removal of the internal organs, the chef meticulously cleans the remaining non-toxic parts of the fish. This involves thoroughly washing the muscle tissue to remove any residual blood or potential contaminants. Some chefs use a fine brush to ensure every surface is pristine. The muscle blocks are then carefully filleted, often yielding translucent, pristine slabs of white flesh. These fillets are then prepared for various dishes. For Fugu sashi, the raw sashimi, the chef's knife skills reach their zenith. The filleted flesh is sliced with breathtaking thinness, often so delicate that the pattern of the plate can be seen through the translucent slivers. This technique, known as usu-zukuri, requires a knife of exceptional sharpness and a hand of unwavering steadiness. The slices are then artfully arranged on a platter, often in an intricate design, sometimes resembling a chrysanthemum, a symbol of longevity and beauty in Japanese culture. The presentation itself is a form of edible art, reflecting the chef's aesthetic sensibility and profound respect for the ingredient. The chef's knowledge extends beyond mere anatomy and knife skills. It encompasses an understanding of the different species of pufferfish, their varying levels of toxicity, and even seasonal changes in toxin concentration. They must know which parts of which species are edible and which are absolutely forbidden. This deep, nuanced understanding is not learned from a textbook alone. It is absorbed through years of direct experience, observation, and mentorship under seasoned masters. The philosophical undercurrent of the Fugu chef's work is equally profound. There is an acceptance of the inherent danger not as a thrill, but as a responsibility. The chef is a guardian protecting the diner from the very poison that makes the fish so unique. This requires a strong moral compass, an ethical commitment that transcends mere culinary ambition. The trust placed in the chef by each diner is immense, a silent pact that the chef will exercise their skill and knowledge with absolute, unwavering diligence. This arduous journey, this dedication to precision, transforms the Fugu chef from a cook into a true artisan, a guardian of a dangerous tradition. Their hands, guided by years of practice and a profound respect for the potent secrets of the ocean, become instruments of both art and safety. The blade's edge, razor-sharp and unwavering, is not just a tool for cutting. It is a symbol of the chef's mastery, a testament to their unwavering commitment to the way of the Fugu, a path where every cut, every decision carries the weight of life and stillness. Guardians of the palate, the rigorous path to Fugu mastery. From the precise, almost surgical art of the Fugu chef, our focus now gently widens to encompass the entire system that cultivates such extraordinary mastery and ensures the safety of those who partake in this unique culinary tradition. This brings us to the rigorous, almost legendary path to Fugu licensure in Japan, a process designed to create true guardians of the palate. Individuals whose expertise is not merely culinary, but also deeply scientific and profoundly ethical. It is a system built on meticulous training, stringent examinations, and an unwavering commitment to preventing even a single tragic misstep. Imagine a young apprentice drawn to the mystique and challenge of Fugu, stepping into a licensed Fugu restaurant for the very first time. Their journey will be long, often spanning many years, typically between 5 to 10, before they can even contemplate sitting for the official licensure examination. This is not a fast track. It is a slow, immersive process of observation repetition and deep learning, akin to the quiet, steady growth of a centuries-old tree. The apprenticeship begins with the most fundamental tasks, far removed from the delicate handling of the Fugu itself. It might involve washing dishes, cleaning the kitchen, preparing vegetables, and observing the seasoned chefs with an intense, silent focus. This initial phase is crucial for instilling discipline, humility, and an understanding of the meticulous hygiene and order required in a Fugu establishment. The apprentice learns the rhythm of the kitchen, the subtle cues and the unspoken rules that govern this sacred space. As time progresses, the apprentice gradually moves closer to the Fugu itself, but still under strict supervision. They might be tasked with cleaning the non-toxic parts of the fish, learning to identify different species, and understanding the nuances of Fugu anatomy. They observe their master chef, watching every cut, every gesture, every decision, absorbing the wisdom through silent observation and diligent practice. This period is not about speed, but about developing a profound respect for the ingredient and an intuitive understanding of its inherent danger. The theoretical knowledge required is immense. A Fugu chef must possess an encyclopedic understanding of the various species of pufferfish, including their scientific names, geographical habitats, seasonal variations in toxicity, and the precise distribution of tetrodotoxin within their bodies. They must be intimately familiar with the chemical properties of tetrodotoxin itself, its mechanism of action, and the symptoms of poisoning. This scientific grounding is as critical as their knife skills, providing the intellectual framework for their practical decisions. Once the apprentice has accumulated sufficient experience and their master deems them ready, they may apply for the prefecture-level Fugu handling examination. This examination is notoriously difficult, with pass rates often hovering around 30–50%, sometimes even lower. It is a multi-stage assessment designed to rigorously test both theoretical knowledge and practical skill. The written examination is a formidable hurdle delving deep into Fugu biology, toxicology-relevant laws and regulations, and public health standards. Candidates must demonstrate not just rote memorization, but a profound understanding of the subject matter, showing they can apply their knowledge to complex scenarios. Imagine a quiet examination hall filled with the focused intensity of individuals who have dedicated years of their lives to this moment, their pens moving slowly, deliberately across the paper, each answer a testament to their long journey. The practical examination is where the true test of precision and nerve unfolds. Each candidate is presented with a live fugu. They are then tasked with preparing it, typically under the watchful eyes of several stern examiners. Every step of the process is scrutinized, the initial stunning, the removal of fins and skin, and most critically, the precise evisceration of the toxic organs. The candidate must demonstrate absolute accuracy in identifying and separating the poisonous parts, ensuring no contamination of the edible flesh. A critical part of the practical exam often involves presenting the prepared, cleaned, edible portions, along with the meticulously separated toxic organs clearly labeled for inspection. The examiners will carefully check for any traces of edible flesh on the toxic parts, or conversely, any remnants of toxic material on the edible portions. A single speck of liver tissue on a fillet, a tiny piece of ovary clinging to the skin, would result in immediate failure. This uncompromising standard reflects the zero-tolerance policy for error when dealing with such a potent toxin. In some prefectures, an additional nerve-wracking component of the practical exam involves the candidate consuming a small, non-lethal portion of the fugu they have just prepared. This is not a test of courage in the face of certain death, but rather a final symbolic act of confidence in their own meticulous skill. A demonstration of their absolute conviction that the fish they have prepared is safe. It is a profound act of trust in their own hands, their own knowledge. Upon successfully passing both the written and practical examinations, the candidate is awarded a Fugu preparation license, a document of immense prestige and responsibility. This license is not a permanent grant. It requires periodic renewal, often involving refresher courses or re-examinations, ensuring that licensed chefs remain current with best practices and maintain their high standards. The existence of this rigorous licensing system has had a dramatic impact on public safety. Prior to the establishment of comprehensive regulations, fatal fugu poisonings were unfortunately common. However, with the implementation of strict licensing and oversight, the number of incidents has significantly decreased, particularly in licensed restaurants. The few incidents that still occur are almost exclusively attributed to amateur preparation by unlicensed individuals or to diners consuming wild-caught fugu prepared outside of regulated channels. Thus, the fugu chef, a guardian of the palate, stands as a testament to humanity's capacity for extraordinary skill, profound dedication, and an unwavering commitment to safety in the face of inherent danger. Their journey is one of silent discipline, meticulous practice, and deep scientific understanding, culminating in a mastery that allows them to transform a creature of quiet lethality into a revered culinary experience. This rigorous path to fugu mastery is a living embodiment of respect. Respect for the fish, respect for the toxin, and ultimately a profound respect for human life. The slow descent, symptoms of Tetrodotoxin, exposure. Having explored the rigorous path to fugu mastery and the meticulous precision required to transform a potentially lethal creature into a culinary delicacy, our contemplation must now, with a quiet solemnity, turn to the other side of the equation, the profound and terrifying experience of Tetrodotoxin poisoning itself. Imagine, if you will, the slow, insidious descent that unfolds when the body encounters this silent architect of stillness, an experience that progresses from subtle whispers of discomfort to a terrifying conscious paralysis. This is not a sudden, violent assault, but rather a gradual, almost gentle unraveling of the body's vital functions, leaving the mind acutely aware of its own terrifying entrapment. The onset of symptoms can be remarkably swift, often appearing within a mere 20 minutes to 3 hours after the ingestion of toxic fugu. though in some rare cases it might be delayed for several hours. This variability depends on the amount of toxin consumed the individual's metabolism, and even the specific species of pufferfish involved. The initial signs are often subtle, almost innocuous, easily mistaken for mild indigestion or a passing discomfort, yet they are the first faint whispers of the toxin's pervasive reach. One of the very first sensations a person might experience is a peculiar tingling or numbness, often described as paresthesia, that begins around the mouth and lips. Imagine a faint pins and needles sensation, a subtle prickling that spreads gently across the facial area, perhaps extending to the tongue. This is the Tetrodotoxin beginning its work, subtly interfering with the sodium channels in the sensory nerve endings of the oral cavity. It is a quiet, almost playful sensation, yet it carries a profound and ominous significance. As time progresses, this tingling sensation typically spreads, reaching the fingers and toes, then gradually moving inwards to the extremities. A growing numbness settles over the limbs, accompanied by a subtle weakness. Imagine the feeling of your hands becoming heavy, your fingers less responsive, as if they are no longer entirely your own. Simple tasks like holding a utensil or lifting a glass become increasingly difficult, demanding a conscious effort that was previously effortless. This is the toxin methodically shutting down the electrical pathways in the motor nerves, gently diminishing the body's ability to respond to the brain's commands. Gastrointestinal symptoms frequently accompany these initial neurological signs. Nausea may begin to churn gently in the stomach, followed by vomiting, often forceful and persistent, as the body attempts to purge the ingested poison. Diarrhea can also occur, adding to the body's distress and fluid loss. These visceral reactions are the body's desperate, albeit often insufficient attempt to expel the harmful substance, a last-ditch effort before the toxin's deeper effects take hold. As the paralysis deepens, the victim's coordination begins to falter. The ability to walk steadily to maintain balance becomes compromised. They may stumble, their gait becoming unsteady, as if walking on an unseen shifting surface. This ataxia, or loss of coordination, is a clear indication that the nervous system's intricate control over movement is being progressively disrupted. The muscles, though not yet fully paralyzed, are losing their finely tuned ability to work in concert. A more distressing symptom that often emerges is dysarthria. Difficulty in speaking. The muscles of the tongue, lips, and throat, which are essential for articulating words, begin to weaken. Speech becomes slurred, indistinct, the words forming with great effort, sounding thick and heavy. Imagine the frustration, the growing terror, as the ability to communicate, to cry for help, slowly slips away, replaced by garbled sounds or complete silence. This loss of voice is a profound and isolating experience, trapping the individual within their own failing body. The eyes, those windows to the soul, also fall under the toxin's influence. Mydriasis. The dilation of the pupils may occur, making them appear wide and unreactive to light. The muscles controlling eye movement can also be affected, leading to diplopia, or double vision, as the eyes struggle to coordinate their gaze. Imagine the world blurring, becoming indistinct, as the visual input itself becomes distorted, adding to the disorienting and frightening experience. As the tetrodotoxin continues its insidious work, the paralysis deepens, moving from the extremities and face towards the core of the body. The muscles of the neck and trunk become heavy, unresponsive. The ability to move limbs, to shift position, to even hold one's head upright diminishes. The victim becomes increasingly immobile, sinking into a state of flaccid paralysis. This is the terrifying threshold where the body, though still alive, begins to lose its capacity for independent action, becoming a heavy, unresponsive shell. The most critical and life-threatening stage of tetrodotoxin poisoning is the onset of respiratory paralysis. The diaphragm, the primary muscle responsible for drawing breath, and the intercostal muscles which aid in expanding the chest cavity, are composed of skeletal muscle fibers that are highly dependent on nerve impulses from the brain to contract. As the sodium channels in the motor nerves leading to these vital muscles are blocked, they cease to function. Breathing becomes shallower, more labored, until it eventually ceases altogether. Imagine the desperate conscious struggle for air, the terrifying realization that the body can no longer perform the most fundamental act of life. This respiratory arrest is the ultimate cause of death, often occurring within 4 to 6 hours after ingestion, though it can be sooner or later depending on the dose. Throughout this horrifying progression, a crucial and particularly cruel aspect of tetrodotoxin poisoning is that consciousness often remains intact. The blood-brain barrier, while not entirely impenetrable, significantly slows the toxin's access to the central nervous system compared to its rapid assault on peripheral nerves. This means that for a significant period, the victim remains fully aware of their surroundings, fully conscious of their own body's terrifying descent into paralysis. They can hear, they can see, they can understand, but they cannot move, cannot speak, cannot signal for help. It is a state of conscious entrapment, a silent scream from within an unresponsive vessel, a profound psychological torment superimposed upon the physical incapacitation. The heart, while also affected, is often more resistant to tetrodotoxin than the respiratory muscles due to its intrinsic pacemaker cells and different types of sodium channels. However, at higher doses, or as the body succumbs to oxygen deprivation from respiratory failure, cardiac arrhythmias, and eventually cardiac arrest can occur. But typically, it is the cessation of breathing that claims life, first leaving the heart, to beat for a short, agonizing time, in a body that can no longer draw the breath of life. The slow descent into tetrodotoxin poisoning is a harrowing journey, a testament to the profound power of a single molecule to unravel the delicate symphony of life. It is a reminder of the immense respect owed to the silent secrets of nature and the critical importance of the guardians of the palate who stand between this potent danger and the trusting diner. The symptoms paint a vivid, chilling picture of a conscious mind trapped within a body slowly rendered still, a profound silence descending upon the very essence of being. Echoes from the past. Tales of Fugu's fatal embrace. Having contemplated the chilling step-by-step unraveling of life under the influence of tetrodotoxin, our journey now turns to the solemn echoes from the past, to the historical accounts and poignant narratives that underscore the profound danger inherent in Fugu. These are not mere statistics, but stories that speak of human curiosity, culinary daring, and the unforgiving power of nature's silent poisons. They serve as stark reminders of why the rigorous path to Fugu mastery and the strict regulations surrounding its preparation are not merely traditions, but absolute necessities. The history of Fugu consumption, as we've briefly touched upon, stretches back through the mists of time in Japan, evidenced by ancient shell mounds containing pufferfish bones. This long history is inevitably intertwined with accounts of accidental poisonings, whispers of those who succumbed to the fish's lethal embrace. Before the advent of modern medical understanding and stringent governmental regulations, Fugu fatalities were a tragically common occurrence, a grim toll extracted by the allure of this dangerous delicacy. Imagine a time centuries ago in a quiet fishing village along the Japanese coast. A fisherman perhaps brings home a pufferfish, its plump form promising a hearty meal. Unaware of the precise distribution of the toxin, or perhaps overconfident in traditional, less scientific methods of preparation, the fish is cleaned and cooked. The family gathers eager for the meal. The initial tingling sensations might be dismissed as a peculiar flavor, a novel experience. But then, the numbness deepens, the limbs grow heavy, the breath falters. The scene unfolds not in a sterile hospital, but in a humble home, surrounded by loved ones who are helpless witnesses to a slow, agonizing descent into stillness. These were the unrecorded tragedies, the private sorrows that shaped a collective understanding of Fugu's potent danger. One of the most widely cited and poignant historical incidents occurred in 1888, involving the then Prime Minister of Japan, Ito Hirobumi. While not a direct fatality, the story highlights the pervasive danger. During a visit to Shimonoseki, a city renowned for its fugu, Ito Hirobumi was served the fish. Despite a long-standing ban on fugu consumption imposed due to previous fatalities. Ito was so impressed by the taste that he lifted the ban in that specific prefecture. This act, while perhaps a testament to fugu's irresistible allure, also underscores the constant tension between enjoyment and danger, and the historical struggle to control its consumption. Another well-documented tragedy involved the renowned kabuki actor Bando Mitsugoro VIII in 1975. A celebrated figure on the Japanese stage, Mitsugoro dined at a Kyoto restaurant and, despite warnings from the chef, insisted on being served 4 portions of fugu liver. The liver, as we know, is one of the most toxic parts of the fish. He reportedly consumed it, stating he enjoyed the exquisite taste. Within hours, he experienced the classic symptoms: paralysis, difficulty breathing, and tragically, he succumbed to the Tetrodotoxin. This incident sent shockwaves through Japan and served as a stark public reminder that even in an era of licensed chefs, individual choices and the inherent potency of the toxin could still lead to fatal consequences. It reinforced the strict prohibition on serving fugu liver, which remains illegal even for licensed chefs due to its extreme toxicity. Beyond these prominent historical figures, countless other less-known individuals have over the centuries fallen victim to fugu poisoning. Many of these incidents, particularly in more recent times, are not linked to licensed restaurants, but rather to amateur attempts at preparing the fish at home, or to fishermen consuming their own catch without proper knowledge or training. Imagine the quiet confidence of someone who believes they can handle the fish, perhaps having seen it done or relying on incomplete information. The outcome tragically often mirrors the slow descent we previously described, a private struggle culminating in a silent stillness. The Japanese Ministry of Health, Labor and Welfare maintains meticulous records of fugu poisoning incidents. While the number of fatalities has significantly decreased since the implementation of the strict licensing system in 1958, incidents still occur almost annually. The vast majority of these cases consistently involve individuals who have caught pufferfish themselves and attempted to prepare it at home, or consumed it from unlicensed sources. This pattern powerfully demonstrates the efficacy of the licensing system in preventing deaths in commercial settings, and simultaneously highlights the enduring danger when that expertise is absent. These records paint a consistent picture of the progression of symptoms, the initial tingling, the spreading numbness, the gastrointestinal distress, the increasing difficulty in movement and speech, and finally, the respiratory, paralysis. The stories, whether ancient or modern, are variations on a single, terrifying theme, a conscious mind trapped within a body that has ceased to obey, a silent witness to its own fading life force. The tales of Fugu's fatal embrace are not merely cautionary. They are also a profound testament to the power of human ingenuity and the evolution of cultural practices. The very existence of a thriving Fugu industry operating safely under strict regulations is a direct response to these historical tragedies. It is a testament to the meticulousness of Japanese culinary tradition, which has acknowledged the danger, understood its mechanism, and developed a system to mitigate the risk, transforming a historical threat into a controlled, revered experience. These echoes from the past, these stories of both fascination and tragedy ripple through time, reminding us of the delicate balance between humanity and the potent forces of the natural world. They whisper of the profound respect owed to creatures like the pufferfish, whose silent chemical secret has shaped culinary practices, inspired rigorous training, and left an indelible mark on the cultural and medical history of Japan. They are stories that underscore the enduring power of Tetrodotoxin and the vital importance of the guardians who stand between its silent, deadly embrace and those who seek to experience the unique allure of Fugu. Beyond the bite, the medical enigma of Tetrodotoxin. Having journeyed through the historical shadows of Fugu's fatal embrace and the poignant tales that underline its profound danger, our contemplation now shifts to a different realm, the intricate world of medicine and pharmacology. Here, Tetrodotoxin, that silent architect of stillness, transcends its role as a mere poison and emerges as a fascinating medical enigma, a molecule whose precise mechanism of action offers tantalizing possibilities for therapeutic applications. The very potency that makes it so lethal also makes it a subject of intense scientific scrutiny, a key that could unlock new understandings of pain, nerve function, and even the regeneration of damaged neural pathways. Imagine, if you will, the profound paradox: a substance so devastating in its natural form, yet holding the potential for healing. This paradox lies at the heart of Tetrodotoxin's medical intrigue. Its exquisite specificity in blocking voltage-gated sodium channels is precisely what makes it so valuable to researchers. In the laboratory, purified Tetrodotoxin acts as an indispensable tool for neuroscientists, allowing them to precisely manipulate nerve activity. By selectively blocking sodium channels, scientists can study the role of these channels in various physiological processes, dissecting the intricate dance of electrical signals that orchestrate life. It's like having a master switch that can selectively silence specific parts of a complex electrical circuit, allowing for a deeper understanding of each component's function. One of the most promising areas of research revolves around Tetrodotoxin's potential as an analgesic, a powerful pain reliever. Chronic pain, a relentless and debilitating condition, often stems from the overactivity of sodium channels in nerve cells, which leads to persistent, unwanted pain signals. Because Tetrodotoxin can effectively silence these channels, scientists theorize that it could potentially offer a novel approach to pain management, particularly for severe, intractable pain that does not respond to conventional treatments. Imagine a medication derived from this potent toxin, carefully engineered to target only the pain-transmitting nerves, offering profound relief without the systemic side effects of current opioids. Clinical trials have explored the use of extremely low, carefully controlled doses of Tetrodotoxin for various forms of neuropathic pain, including cancer-related pain and phantom limb pain. These studies are conducted with the utmost caution, meticulously monitoring patients for any signs of toxicity. The hope is to find a therapeutic window where the analgesic effects are achieved without inducing the dangerous paralysis associated with higher doses. The challenge lies in precisely controlling the dosage and delivery method, ensuring that the toxin acts locally or in a highly targeted manner rather than spreading throughout the nervous system. Beyond pain relief, Tetrodotoxin's unique properties have also sparked interest in its potential role in treating drug addiction, particularly opioid dependence. Scientists believe that by modulating nerve activity, TDX might help to alleviate the severe withdrawal symptoms that make it so difficult for individuals to break free from opioid addiction. The precise mechanisms are still under investigation, but the idea is that by gently dampening the hyperactive neural pathways associated with withdrawal, TTX could provide a smoother, less agonizing detoxification process. This is a complex area of research fraught with challenges, but the potential to offer new hope to those struggling with addiction is a powerful motivator. Another intriguing, though highly speculative, area of research involves nerve regeneration. Some preliminary studies have hinted that Tetrodotoxin, by temporarily silencing nerve activity, might create an environment conducive to nerve repair or regeneration after injury. Imagine a damaged nerve constantly firing chaotic signals, unable to properly heal. By temporarily quieting this chaotic activity, TTX might allow the nerve to rest, reorganize, and potentially begin the process of repair. This is a frontier of neuroscience still in its nascent stages, but it underscores the sheer breadth of possibilities that a molecule with such precise biological activity can present. The medical applications of Tetrodotoxin are not without significant challenges. The primary obstacle, of course, is its extreme toxicity. Any therapeutic use requires incredibly precise dosing and controlled delivery systems to ensure that only the desired effects are achieved without crossing into dangerous territory. Research involves developing highly purified forms of TTX, exploring novel drug delivery methods such as localized injections or slow-release formulations, and understanding the exact pharmacokinetics and pharmacodynamics of the toxin in the human body. The margin between a therapeutic dose and a toxic dose is incredibly narrow, demanding meticulous scientific rigor and ethical oversight. Furthermore, the source of Tetrodotoxin for medical research is also a consideration. While it can be extracted from pufferfish, this is not a sustainable or scalable method for pharmaceutical production. Scientists are actively working on synthetic pathways to produce Tetrodotoxin in the laboratory, ensuring a consistent, pure, and ethically sourced supply for research and potential medical use. This synthetic approach would also allow for the creation of analogues, slightly modified versions of the molecule that might retain the desired therapeutic properties while having a reduced toxicity profile. The journey from a deadly natural poison to a potential life-saving medication is long and arduous, paved with countless experiments, rigorous clinical trials, and an unwavering commitment to safety. Tetrodotoxin in its natural form remains a silent killer, a stark reminder of nature's power. Yet in the hands of dedicated scientists and medical professionals, it transforms into a medical enigma, a molecule whose profound ability to silence nerve signals could one day offer solace to those suffering from intractable pain, or even pave the way for new treatments for neurological disorders. So, as we consider Tetrodotoxin, let us look beyond the bite, beyond the immediate danger, and contemplate the deeper scientific mysteries it holds. It is a molecule that whispers of the intricate connections between life and stillness, between poison and cure. inviting us to explore the profound and often surprising ways in which nature's most potent secrets can be harnessed for the benefit of humanity. The medical enigma of Tetrodotoxin is a testament to the enduring human quest to understand, to innovate, and to ultimately alleviate suffering, even by carefully embracing the very essence of danger. Diversity in danger, the global family of pufferfish. Having ventured into the profound medical enigma that Tetrodotoxin presents, our journey now gently expands beyond the immediate focus of Japanese Fugu to embrace the vast and diverse global family of pufferfish. These intriguing creatures, united by their remarkable ability to inflate and their shared chemical secret, populate an astonishing array of aquatic environments across the planet. Each species a unique testament to the power of evolution and the widespread distribution of danger in the natural world. The family. Tetraodontidae is a large and varied one encompassing over 120 recognized species, each with its own subtle variations in appearance, habitat, and crucially, toxicity. Imagine drifting through the warm, crystal-clear waters of a Caribbean coral reef. Here you might encounter species like the band-tail puffer, Spheroid spengleri. Its body adorned with subtle bands and speckles perfectly camouflaged against the sandy seafloor. Or perhaps the southern puffer, Spheroides nephilis, with its gentle eyes and soft, rounded form, grazing peacefully on algae. These tropical species, while perhaps not as famous as their Japanese cousins, are nonetheless bearers of Tetrodotoxin, their presence a silent warning to predators and unwary humans alike. Their toxicity levels can vary, but the potential for harm remains a constant, inherent part of their being. Travel northward into the temperate waters of the Atlantic and Pacific, and you will find other members of this diverse family. The northern puffer, Spheroides maculatus, for instance, is a common sight along the eastern coast of North America. Known colloquially as sugar toads, in some regions these puffers are often caught by recreational fishermen, while their flesh is considered edible and is even commercially harvested in some areas. Meticulous cleaning is still absolutely essential, as their internal organs, particularly the liver and gonads, can still harbor dangerous levels of tetrodotoxin. This underscores a crucial point. Not all pufferfish species are equally toxic, and even within a single species, toxicity can vary based on geographical location, diet, and season. The Indo-Pacific region, with its sprawling coral reefs and nutrient-rich waters, is a particularly vibrant hotspot for pufferfish diversity. Here you might encounter the striking starry puffer, Erythron stellatus, a large, majestic species covered in white spots. Its slow, deliberate movements a captivating sight amidst the coral gardens. Or the intricate patterns of the map puffer, Arothron mappa, its skin resembling an ancient hand-drawn chart. These larger, often more visually dramatic species are frequently highly toxic, their beauty concealing a potent chemical defense. Divers and snorkelers often observe them with a mixture of awe and respectful distance, recognizing their inherent danger. Beyond the marine realm, some species of pufferfish have adapted to brackish water, where fresh and salt water mingle in estuaries and mangrove swamps. A few remarkable species have even made the complete transition to freshwater environments, living exclusively in rivers and lakes. The dwarf puffer, Carinotetraodon travancoricus, a tiny, enchanting species from India, and various species of Tetraodon found in African and Asian rivers are popular aquarium fish. While many freshwater puffers are considered less toxic than their marine counterparts, some can still produce or accumulate tetrodotoxin, albeit often at lower concentrations. The green-spotted puffer. Tetraodon nigroviridis. For example, a popular brackish water aquarium inhabitant is known to be toxic. This wide ecological distribution further illustrates the adaptability of the pufferfish family and the pervasive nature of their chemical defense. The variety in appearance among pufferfish is as striking as their geographical distribution. Some are smooth-skinned, while others are covered in small, prickly spines that become erect when the fish inflates. Their coloration ranges from subtle camouflaged browns and greys to vibrant, almost psychedelic patterns of spot stripes and blotches. Their sizes vary dramatically from the tiny fingernail-sized dwarf puffers to large, imposing species that can grow to over a meter in length. Yet beneath this kaleidoscope of forms, the shared characteristics of their fused beak-like teeth, their inflatable bodies, and their potential for tetrodotoxin toxicity bind them together as a distinct and fascinating family. The scientific understanding of this global diversity in danger is crucial for public health, particularly in regions where pufferfish are consumed. While Japan has the most stringent regulations, other countries in Southeast Asia, such as Korea, China, and various island nations also have traditions of consuming pufferfish. The challenge in these regions is often the sheer number of species, the varying local names, and the difficulty in distinguishing highly toxic species from less toxic ones, or even from non-toxic fish that may resemble puffers. This can lead to tragic incidents, particularly in areas where regulations are less strict or where individuals attempt to prepare the fish without proper training. Moreover, the phenomenon of climate change and ocean warming is beginning to introduce new complexities. As marine species shift their geographical ranges in response to changing environmental conditions, toxic pufferfish species may appear in waters where they were previously uncommon. This poses a new challenge for local fisheries and public health officials, who must continuously monitor and adapt to these evolving ecological dynamics, ensuring that communities are aware of the potential dangers of newly arriving species. The global family of pufferfish therefore represents a profound example of diversity in danger. Each species, whether gliding through tropical reefs, sifting through temperate sands, or navigating brackish estuaries, carries within its unassuming form a silent, potent secret. They are a testament to the intricate and often surprising ways in which nature equips its creatures for survival, reminding us that beauty and peril can often reside in the same delicate package. Their widespread presence across the world's waters serves as a constant, gentle reminder of the ocean's boundless mysteries and the enduring respect we must hold for its powerful, often unseen forces. The ocean's paradox: beauty, peril, and enduring fascination. As our contemplative journey through the world of the pufferfish and its potent secret gently approaches its horizon, we are left with a profound sense of wonder, a quiet appreciation for the intricate paradoxes woven into the very fabric of our natural world. The pufferfish, in its myriad forms and habitats across the globe, stands as a living embodiment of the ocean's enduring mystery, a creature of subtle beauty, yet simultaneously a bearer of silent peril, forever holding humanity in its enduring fascination. Imagine one last time a pufferfish, drifting serenely amidst a vibrant coral reef, its large, expressive eyes scanning the kaleidoscopic environment. Its movements are unhurried, almost meditative, a testament to a life lived in harmony with the gentle undulations of the currents. Its form, whether smooth or subtly spined, is a marvel of evolutionary design, capable of astonishing transformation from a streamlined swimmer to an impenetrable spiny sphere. There is an undeniable elegance in its very existence, a quiet dignity that captivates the observer, drawing the eye to its unique patterns and its almost contemplative gaze. This beauty, often understated, is a reminder of the endless artistry that nature expresses in every living form, even those that harbor potent secrets. Yet beneath this calm exterior, within the very tissues of its being, lies the silent architect of stillness, Tetrodotoxin. This potent neurotoxin, acquired from microscopic bacterial alchemists through complex food webs, transforms the pufferfish from a mere inhabitant of the ocean into a formidable guardian, a living declaration of its own inviolability. It is a chemical shield meticulously refined over millions of years that renders the pufferfish almost untouchable, a testament to the elegant brutality of natural selection. This inherent peril, this capacity for swift and silent lethality, is a profound aspect of its fascination, drawing our minds to the delicate balance between life and stillness, between curiosity and caution. The human response to this paradox has been as complex and varied as the pufferfish itself. In Japan, particularly, the challenge of Fugu has been embraced not with fear, but with a profound respect and an extraordinary dedication to mastery. The rigorous path of the Fugu chef, demanding years of intense training, meticulous precision, and an unwavering ethical commitment, stands as a testament to humanity's capacity to engage with danger, to understand its mechanisms, and to transform it through skill and knowledge into a revered culinary art. It is a cultural phenomenon that speaks volumes about the human spirit's desire to push boundaries, to seek unique experiences, and to honor the intricate dance between risk and reward. The historical echoes of Fugu's fatal embrace, those poignant tales of accidental poisoning, serve as solemn reminders of the toxin's unforgiving power. They underscore the critical importance of knowledge regulation and respect when interacting with such potent natural forces. These stories are not just cautionary, they are also integral to the ongoing narrative of human adaptation and learning, shaping the very systems that now allow Fugu to be enjoyed safely in licensed establishments, a controlled dance on the edge of a precipice. Beyond its culinary and historical significance, Tetrodotoxin continues to intrigue the medical and scientific communities. Its precise mechanism of action, its ability to selectively silence specific sodium channels, positions it as a powerful tool for neuroscience research, offering tantalizing possibilities for new therapeutic applications. Imagine the potential: a substance derived from a deadly poison, carefully harnessed to alleviate chronic pain, to treat addiction, or even to aid in nerve regeneration. This transformation from lethal agent to potential medicine is a testament to the relentless human quest for understanding, for turning nature's secrets into solutions for suffering. The global family of pufferfish, with its astonishing diversity in appearance, habitat, and toxicity, further deepens our appreciation for this ocean paradox. From the vibrant reefs of the Indo-Pacific to the temperate coasts of the Atlantic, from brackish estuaries to freshwater rivers, these creatures thrive, each carrying its unique blend of beauty and danger. Their widespread presence reminds us that the ocean, in its vastness and complexity, is a realm where life and death are intimately intertwined, where fragility and formidable power often reside in the same unassuming form. And so, the pufferfish remains a silent, enduring symbol of the ocean's profound paradox. It is a creature that invites us to contemplate the delicate balance of ecosystems, the hidden chemical pathways of life, and the intricate relationship between humanity and the natural world. Its story is one of beauty that conceals peril, of danger that inspires mastery, and of a quiet, persistent fascination that continues to ripple through our scientific endeavors and our cultural practices. The pufferfish reminds us that even in the most serene corners of our planet's vast blue waters, profound mysteries and astonishing powers patiently await our gentle, respectful contemplation. And so as we gently drift away from the profound paradoxes of the pufferfish, its subtle beauty and its potent silent secrets, let us now allow the deep, rhythmic breathing of the ocean itself to wash over us. Feel its vast, tranquil expanse drawing you in, softening the edges of your waking thoughts, dissolving any lingering concerns into the gentle, ebbing tide of your consciousness. Just as the deep currents cradle these enigmatic creatures in their watery realm, let them now tenderly carry your mind, guiding it away from the surface of the day and down into the serene, quiet depths within. The mysteries of the ocean, much like the untold stories that unfold within our dreams, invite a profound surrender to the unknown, a peaceful trust in the unseen forces that guide us through the night. Feel the slow, steady rhythm of your own breath, a gentle wave ebbing and flowing, drawing you deeper into a profound and peaceful stillness. Let your mind become as vast and calm as the ocean at its most tranquil depths, where all is quiet and rest is absolute. May the deep, gentle currents of the dreaming ocean cradle you, safely ensuring your slumber is profound and undisturbed. Sleep well. Goodbye, and see you again.